it is said to avoid anything, side effects of this hangover is to remain intoxicated. And I think NMR is one of the things which the talks can make you intoxicated for a longer time, so that you can avoid the hangovers which you might have last evening. And we have two talks. First one is Krishna Mohan. Mohan is currently at IIT Roorkee. He has done his PhD at the Institute of Fundamental Research, and then he moved to Rutgers for the postdoctoral and University of Texas. Since then, after he returned back to IIT Roorkee, and where he probably the first one in IIT Roorkee to start protein NMR work there, and he's doing many kind of work with protein NMR and some part of metabonomics with algae and other things also. And the discovery also is one part of his research work. So with that, I invite Krishna Mohan to start proceeding.
Thank you very much, Neil, for inviting me for this lecture series. So, good morning to all. So, essentially we are going to have the talk which is continuation of yesterday, like sort of we have discussed a little bit in the practical class. We have seen what are the practical aspects, how to do a titration and so forth, what are the do's and what are the don'ts. So, essentially that 1 hour helped me so that I can correlate what I am talking today. We are going to correlate with what we did yesterday. Okay. So let's start. So if you see as such as a NMR spectroscopy or something, so we have so many of the techniques which can be used to understand the protein ligand interactions. So when we are talking about protein ligand, okay, or ligand doesn't mean it should be a small molecule. It can be anything. Ligand can be other protein, then ligand, if you call ligand as X can be protein, small molecule, DNA, RNA, carbohydrate, anything can be there, right? So we don't differentiate that. So we can use it as a protein-protein interactions, protein nucleic acid interactions, protein carbohydrate interactions, or protein small molecule interactions. So we have several of those. So there are several techniques which of course no one can cover all these techniques in a given time of 1 hour or something. So I will roughly tell what these techniques, few of those are, and I will show you some examples. As the title says, druggable sites are proteins. So I give an emphasis from the sites of proteins. So there are some important aspects like we have discussed yesterday. The way that, if you want to have a simple binding constant, why do I need to do a NMR experiment as such? Okay. We have several of these experiments. I will show you a few of those, and there are some experiments which are more particular to the ligands or the small molecules, where you don't label the protein at all. You will use a one-dimensional NMR spectroscopy, and there are some experiments where you do a 15N sample or a 13C sample, or in some of the experiments where even you deuterate the samples. Okay. So as per the need, we use the labeling schemes or the experiments. So, and different experiments of course will give a lot of variety of information which we will get. Suppose if I say simply a chemical shift perturbation, okay. So yesterday what you have seen is you measured a proton 15N HSQC and you have seen how the peak shifts, right. But as such, if I talk about a chemical shift perturbation, I can talk about proton chemical shifts, 15N chemical shifts, I can talk about C alpha chemical shifts, H alpha chemical shifts, or even methyl proton chemical shifts. But the whole term comes as a chemical shift itself. Okay. So you can also measure some excellent properties using NMR spectroscopy other than a simple binding or something. Okay. the proteins do have conformational changes, so we can measure the conformational changes using NMR spectroscopy. So what the other important feature which is very unique to NMR spectroscopy and what NMR spectroscopy contributes to is the dynamics of the molecule. So what happens to the dynamics of a molecule when a ligand is bound to it? So whether the dynamic ventures, enhances, and the thing is what happens to the stability of the protein? When it is a surface binding or something, it may or may not enhance the stability, or a ligand binding can enhance the stability. Similarly, you can talk about cooperativity, allosteric, and all those features you can talk about, and also you can exactly identify the locations. That is what is more important compared to a normal global techniques like where you do a CD spectroscopy, fluorescence spectroscopy, or an ITC experiment. So if we start with a simple binding curve, this is what I am telling, like sort of you can get a nice binding curve, you can get a KD value, delta G value, and everything within a half an hour or 1 hour by doing a isothermal titration chemical experiment, right? You will get KD, you will get a stoichiometry, and also you will get delta G, delta H, and the T delta S, everything you will get. Now the same thing what you are seeing here, you can do the same thing by using a NMR. So this is what essentially you have done the titration experiment yesterday. You remember, I told all of you that at least take 2 or 3 points when it is saturating. If at 1:1 or 1:2, if a ligand is saturating, I told go for 1:4, 1:6 or something. This is what the reason I told you. See, it is almost saturating here, but you need more points to get a flattened curve in order to fit it. So you can get the thermodynamic properties, that means you can get the KD and everything using NMR spectroscopy. Okay. What else we need when a binding is happening? Yesterday we discussed there are two things, right? One is thermodynamics. You need your delta G, stoichiometry and everything. Second thing is kinetics. So K on and K off. When a ligand is coming and binding to a molecule, you will have a K on, K off, right? So that also, to be frank, do we need a NMR? No, we can do it a K on and K off using another technique which is called the surface plasma resonance. Okay. So, but when we are talking about all these things, there are several features which we miss. That is, none of these techniques, they will give energetic features or the kinetic features, but we don't know the location of binding. We don't know the involvement of which atoms or which amino acids and everything. So what is unique about understanding protein ligand interactions or protein interactions by using NMR spectroscopy is you get all the information what you get from these tools. Additionally, you get lot more information. That is why whoever does NMR spectroscopy, essentially they try to look everything using NMR spectroscopy, since they anyway get what the information is coming from other techniques. But it is always wise to get the information from other techniques to support your NMR data. Okay. So coming back to this, if you have A and B, okay. So now we will discuss 3-4 very important points when we are doing a binding. Think of very simple, one example, like think of you have a protein and you have a ligand. Okay. Now the rest of the 45 minutes or 50 minutes, whatever the talk is, we stick to a protein and ligand and try to analyze what all the different scenarios happens when you have a protein and ligand. Okay. I will tell you few scenarios. Now the ligand can induce stability to the protein. How do you monitor this? Or the ligand can quench the dynamics of a protein. And if you have a heterogeneity in system, that means if you have a major minor conformation or monomer dimer, monomer tetramer or something, there can be ligand induced changes, oligomerization or dissociation happens. A dimer can dissociate into monomers or monomers dissociate into monomers associate into dimers. So that we can see. And then if you take the ligands, okay, what is the guarantee that ligand should bind at one place? It can bind at more places. Then if you have more binding sites on a protein to a certain extent overlapping, how does the chemical shift look like? Okay. So, and at the same time, since we are looking on the druggable surfaces on the proteins, how do these surfaces evolve? How do we identify such surfaces? So there is a some point coming from the NMR, and we have to give such emphasis from the protein side also, right? When we are studying about the protein, we need to understand how these protein surfaces evolve, and if there are protein-protein interactions or something, what are the important interactions you should look at using NMR spectroscopy? First, that's why you should know about the protein. If you know whether I should see a hydrogen bonding or electrostatic interaction, then only as an NMR spectroscopist I can visualize what I should look at, right? So these are essentially the concepts which we are looking for the next 45 minutes. So when you have a NMR binding happens, okay. So I will come back to this. This is how you see when you have a shifting, okay. So that means you have a bound state and unbound state. Like the way yesterday we did the experiment, you have a apoprotein. Suppose you have a one resonance here, right? You have one peak here. Now what happens when you go on adding a ligand? Okay. So obviously one thing happens is you are going from a apo state to a bound state, right? But how do you visualize this? So several ways you can imagine, okay. So you have a apo state, you have a bound state, okay. Think of this wall as a apo state and this wall as a bound state, okay. So now it is dependent on the observer. Suppose here one person apo and bound, they are moving so fast like the fan. So what will I see? I neither see the position of the apo nor the position of the bound, but depending upon where the position of a weighted average of this position I can see, right? So this is what we call in terms of fast exchange. That means I have a peak here completely apo and completely bound. Now I don't see two peaks since the exchange at the rate of exchange is much fast, right? So as a result what you see is the peak slowly shifts, and that we put it as a fast exchange, okay. This we see. And the second thing is there may be a other thing that you can monitor both the things. That means a peak, you have a peak for a apo and you have a peak for the bound form, okay. Now there may be a change in the population. Slowly the apo form reduces its population and the bound form increases. When you can able to monitor both of them, which you call as the slow exchange, so then what happens is you will see the apo form decreases and the bound form increases. So there is in between a slow and a fast, which you call it as an intermediate exchange, okay. In that what happens is essentially, like yesterday I told, in the middle of the concentrations or something, all the peaks starts vanishing out, which we call it as traditionally NMR peak broadening, okay. So even when a fast exchange is also happening, suppose when a fast exchange is also happening, you do see such broadening. If you see this is as a fast exchange, you see the size of the circle here and here. The first circle and the last circle are almost equal size, right? But in between we have placed the circles with the circumference as smaller. That means you are losing the peak intensity. That means the peaks start broadening. So we will see these factors also. And you see, now here I had put all these things in a straight line. You may not see them in a straight line, okay. So these are the basics which we use in order to understand how these interactions, when we use real systems of proteins in the next few slides examples when we are showing, we will understand how they look like, okay. So now as I told you, here for these protein ligand interactions we have several techniques, okay. So some of these techniques, if you see the green which I marked, so majorly you try to do from the protein side, you try to monitor from the protein either the relaxation or the hydrogen exchange or some of those cross saturation. I will show few of those examples of those pseudochemical shifts or paramagnetic relaxation, where you use a paramagnetic relaxation depending upon whether the paramagnetic ion is symmetrical or asymmetrical, then you will get a broadening or the shifts which is coming from. So you can use all of this, and one of these very important aspects, surface activity relationship or structure activity relationship, where people use this to identify ligands of much higher affinity, okay. So that means you have a protein, you don't know what ligands to be used. So essentially what we do, you take a database and you start putting all the ligands, right. Then what you get is you get a hit of some ligands. From there, how can I develop from a micromolar binding maybe to a nanomolar binding, which is a high affinity binding. So which comes under the surface activity relationship or structure activity relationship by NMR spectroscopy. So essentially what we are trying to see is, we will see an example for this, and we will also see an example of cross relaxation, hydrogen exchange and chemical shift perturbations. And I briefly touch like STD, since the starting of any experiments is like starting with a 1D, which is STD. And also I have picked up an example in STD where you can see allosteric ligands. Okay. So these are some of the methods which I have taken from some of the review papers. I can share those review papers. So as you see, you cannot get, although you get all the information from NMR spectroscopy, one methodology is not going to give the entire information. That is why I told you, if you see, you have larger proteins or something, you can use SAR NMR, or you can do spin labeling, or you can do STD NMR where you identify the small molecules. So you have different tricks. Okay. Now, the reason why we went to the fast exchange, slow exchange and everything is, again all the experiments won't work under all regimes. This one need to understand. Okay. So that is why we need to know essentially where my binding is. Like when we talk about the protein ligand complexes or protein complexes, like you see, you cannot take a protein and antibody and try to see the K on and K off, right? If something is in a nanomolar, subnanomolar or picomolar, your off rates is almost difficult to get. Similarly, you have transient complexes where the binding is in a micromolar range, right? So depending upon where you are looking at, that is why essentially the initial biophysical experiments will give lot of information to know what exactly you look at NMR. If not, what happens is, the moment you put a sample in NMR, all the peaks broaden, then you say my protein degraded, I don't have to do anything. This is what we try to think. But that is why when you know, that is why people say do ITC, do fluorescence or SPR and see essentially which binding range you are. Then you know precisely what experiments in NMR to do. Okay. So this is one of the thing, saturation difference NMR spectroscopy, which is generally used. So I will tell you how powerful this experiment is, or the NMR people how biased towards this experiment is. Even though you have a fluorimeter next to them, they go to NMR spectrometer and do a STD NMR, they don't touch the fluorimeter. That much favorite, the two experiments which are most favorite is chemical shift perturbations and the STD NMR. So what essentially you do here, so you have a protein and you have a ligands, right? So now what you do is, you selectively irradiate a portion of the protein, which is generally in the range of 1.5 to 3 ppm or something in the methyls. Then you transfer this saturation to the ligand. Now what happens is, the intensity of the ligand changes only when the ligand is bound to the protein. So in a way, what is useful for it? So I don't need to have one ligand. Even if I have 10 ligands, okay, I can put all the 10 ligands and I can screen which one is the best ligand. So this is the specificity of this experiment. It's not like just to get a KD of an experiment or something, right? So out of 10 ligands, I can pick one ligand, since the one which is in K on, K off with the protein, that only will show a differential intensity. The ones which doesn't bind to the protein, like this one, non-ligand, their intensities, when you do a on experiment, on resonance and off resonance experiment, then the difference will be essentially 0. So you can identify this one. Okay. Now this is one good example which I found using the STD NMR experiments. Like first thing what I am telling is, like sort of you have the on and off, you have a ligand. But what you can do is, in the presence of one ligand, you can see how the second ligand binds. This is like a allosteric effect. On one corner, one ligand is present, second ligand. So if you see this one, a ligand is already present. So this is the signal of caffeine. Okay, you have a caffeine signal. Now the binding of glucose to the protein now essentially enhance the signal of caffeine. Okay. Similarly, now you can take caffeine and you can use this compound. In the presence of caffeine, now you add caffeine as a allosteric modulator, then the signal of the compound increases. So how two compounds, you can not only for one compound binding, you can go for multiple compounds and you see the allosteric of this. And if you take these STD NMR sort of experiments, okay, now you see NMR people are very fancy. Okay, today you have one experiment, then I will change couple of pulses and I try to get more information out of it, then I will have a set of experiments. If you see the STD experiment here, very similar to the STD experiment is water loxy. What essentially water loxy and STD differs is, in the water loxy, what you do is, you see the water environment. Since we all know in a protein ligand or protein-protein biomolecular interactions, water is going to play a major role, right? So you will see, you will play with the water around the ligand. Like that, now the pharma companies have used, one is the transferred nosy. Okay, when you take for any small molecule, generally the nosy buildup will be very long and it will be positive in nature. Okay, but for large molecules like proteins or something, the nosy is negative, NOE is negative. So that what you do is you transfer the NOE signal from a protein to the ligand, and the bound ligand now gets a negative signal due to the transfer nosy. So that's how you can see how your ligands are binding. So all these techniques essentially, the reason I kept all these techniques at one place is you don't need any labeling for all these things. So these are the experiments, one dimensional experiments, and anyone can do. Only requirement is you have a spectrometer and a complex system what you have to work with. And in all these things, remember one more thing. So none of these complexes are 1:1 complexes or 1:2 complexes. Okay. So essentially what you do is you take your ligands much more excess. That is where it will settle between the complex and uncomplex states. Okay. And that is one of the very bottleneck. So essentially yesterday we are seeing this one. If you have a complex and if these ligands are only present or only dissolved in DMSO or some of those things, what happens is, due to the hydrophobic nature of these ligands or something, the more the ligand you add, essentially what happens? It starts oligomerizing or precipitating out, and that is one of the biggest bottleneck where these 1:20 or 1:50 sort of experiments don't work practically. Okay. So now this is one more thing. This is nothing to do with your STD NMR, but this example what it tells is you can use any nucleus. Remember, you don't need to go to STD NMR. You can use fluorine, you can use carbon 13, phosphorus 31, any of those you use, you can get an information. Okay. So we use fluorine 19 is one of those which people regularly use. So simple chemical shifts also with heteroatoms, you can use it to identify the things. Since fluorine NMR people label the aromatic amino acids and everything using fluorine, essentially people most of the time use fluorotyrosine. So then you have 4 tyrosines in a protein, essentially I get 4 signals. So life becomes much easier. Okay. So now we will move essentially for the next 30 minutes, the major portion, we will move to the protein part. Okay. The first one is SAR, structure activity relationship, or people say surface activity, since the protein is binding on the pocket on the surface. So what do we do under this? How do we get a high affinity ligand like this? Okay. Essentially the idea is pick up a bag, means whether it is a indole ring, imidazole ring, naphthalene ring, what doesn't matter. First identify, looking at the protein or something, identify some ligand which essentially goes and sits in the pocket. Okay. Rest we will do later. So now here you see, first these are the 2 surfaces. Okay. Screen for the first ligand. So here you have 2 ligands combined. First screen for the first ligand. Now optimize the first ligand, what it comes. Then find, similarly like first ligand, find a second ligand. Now optimize the second ligand. Then you link both the ligands. So now it is not like one ligand has to come, wait, open the site, coming to the second ligand. So both the ligands are coming together. So now if you take this one, this is one of the very standard example of FKBP inhibitors like by SARS NMR. Okay. So now they have these benzene derivatives. Okay. So you can see R's and everything. So they put so many OH groups. So initially they start with this one. Now with all the ones in the first material, what you can have is, so from 2 micromolar, then you can go up to like 0.1 micromolar. So which is already 20 times enhancement, right? Now you take another molecule, okay, this is one unit and this is one unit. Now what you do is, this N is nothing but the linker. Now you increase or decrease the spacing between these linkers, where exactly it fits. Now you play with this. What they found is, when N equal to 3, now one ligand which is a 2 micromolar ending up with a total combination of ligand which is 19 nanomolar. Okay. So 19, if you even take 20, 200, 2. So it's like 100 times much better ligand. So this is what people like to see when they want to do a ligand interactions or something. They like to see in a nanomolar. They want the strongest interactions. And these OH groups, which plays a very major role, how to pick these ligands is essentially, most of the time we go with a very standard naphthalene compounds or benzene compounds or standard organic compounds, or people can start with natural compounds like phenolines, or you can have the flavonoids, terpenoids, some of those. Okay. So this is one thing which we have also actually identified by using this one. So the OH groups plays a major role. So I will show you one of the example which we have seen. Like we have a protein where we identified the flavone ring will sit into it. Okay. Now of course we don't have the chemistry to monitor. So what we did is, went ahead and tried to find naturally occurring chemistry of the molecules where we can use it. So we have taken 3 molecules. You see the rest of the structure ABC rings are same: camphorol, quercetin and myrcetin. The only difference if you see is, one OH is going to 2 OH and 2 OH is going to 3 OH, right? Additional of hydroxy group. And essentially what you see in this is, you will see a much stronger binding due to more interactions of OH groups, and we have seen these interactions. Essentially you can go from a 10 power minus 5, which is like 20 micromolar, to 400 nanomolar. So you can see almost a 50 times stronger binding when you change it. So that means now the more the OH groups, actually the pocket is engaging in more hydrogen bonds. Okay. So now this is the first thing, like you can have structural analysis, SAR technique. Now the thing is, we move to the second point. The second point is, what happens if I have a multiple binding? Okay, this is one of the very known article by Peter Wright, who essentially told, like sort of, what if I see a chemical shift rather than moving straight, it goes to a curved nature or something. So what essentially it tells is, now there are, for the same ligand, they do exist 2 binding sites. Now when we have 2 binding sites for the same ligand, again there is a complication, right? The 2 binding sites can be completely different, or the 2 binding sites can have some overlap. So such curvatures happens when you have a overlap between these 2 binding sites. Okay. If they are completely different, these set of residues are different, these set of residues are different, then you will simply get it as N equal to 2. Okay. That also we have example. Like that is what you will see in a normal spectra. If you have, you will see the straight movement of the peaks, but when you have multiple binding of the ligands, where there is some commonality between the ligand binding and the protein surface, so what essentially you see is, instead of straight, some of the peaks starts curving. So that means essentially the curvature is, it's not only a one bound state and one opposed state. It is much more complex. So in this case here, it is shuttling between one bound state and one opposed state and 2 different bound states. So what you are getting is the average of these things. That is why it won't be straight. Okay. So similarly, we have in one of case, this is the system which I will discuss later. So if you see, there is a binding here, there is a binding here. If you see the chemical shift perturbation around 20 and around 60, there is whichever chemical shift. But now, if you take 2 of these orthologs, that means you take a protein, same protein from a murine system and same protein from a human system. So this is also one thing when we are talking about the druggable nature of the proteins. It is very essential to understand how these protein surfaces actually evolved. So that is a part of it. When you are looking at the complex interactions, there are so many factors. I will at least name some of those factors what you should look at when you are looking at the surfaces, or when you are looking especially at the interface of these proteins. Okay. So now if you see this, in these two bars says like this is the first surface which is common in both of those, but in these rectangles in the bottom protein, they do exist is sort of, these pink balls essentially represent broadening of residues. As I told you yesterday, you can have a chemical shift or the peaks can disappear. Anything can happen when there is a binding. So what essentially is, the first one has only one binding surface and the second one has two binding surfaces. This is something which intrigued us, like this is the first one, like this surface if you see, this is human, this is horse and this is mouse. Okay. So in under the evolution, one side of the protein is essentially positive charge, which helps binding to its cognate molecule, which is the heparin. Heparin contains sulfate groups and negative. In fact, heparin is much more negative than your DNA. That is the most negative molecule what we have, right? So now this is the surface what you are seeing essentially these two. But now under the evolution, the moment you go to humans, now you see there is a surface which is coming on the other side also. That is why in the case of human protein binding, instead of a single, you have multiple binding sites. So that is also important when we are working with different homologs or something. They can have evolution, and you can dig. This is the same. You can dig more with all the several 3 parallel proteins. What we have shown is essentially by using the molecular evolutionary analysis. What we have shown is essentially these residues. What you are doing in human under the evolutionary pressure, these are the positively selected residues. So that means the chemokines which are present in the mouse and human are quite different in terms of binding interaction. So ours has multiple binding modes in the humans compared to that of the primary. So we call this one of the mode as a primoidal surface, the other one is an acquired surface. So one is present in all the along from it is present in Laurasians, it is present in primates, it is present in rodents, everything. But the second one is an acquired surface which is present only in the primates, not at all in any of those. Okay, those are some of those binding surfaces. And important thing I will show you. Sometimes why it is important to find a druggable site on a protein, or if you have a protein. Okay, let me tell you, there are several families of proteins, right? Like you take growth factors, you take chemokines, you take even receptors. I will tell you, one of the biggest problem for a drug discovery and drug thing is how to identify a unique ligand to only to that protein. Majority of the time what happens, majority of the papers even we publish for the sake of publication and everything. The reason why they won't go to the end is, these are essentially specific binding to that protein, but non-specific to the family what you are taking, right? So what happens if I have 50 proteins in that under a given family, and all the 50 starts activating with or inhibiting under a one ligand? So this brings complexity. So that is why choosing some of those ligands is difficult, and at the same time nature also designed some of these proteins where it can have very nice what you talk in terms of single nucleotide polymorphisms, SNPs or something, or many of the time one nucleotide change, your amino acids can change, right? One amino acid to other amino acid. This is one such example where you can have the binding. See, you have a CXL1, CXL2, CXL3. Now if you see this one, L, which is a hydrophobic amino acid, is changing into a R, which is arginine, and now K, which is going to I. Okay, it is essentially a swap between a hydrophobic amino acid and a charged amino acid. Okay, it is not much. In one case, one position has hydrophobic, one position has charge. In the second case, they flip. Okay. Then, in case only CXCl3, the naphthalene derivative can go on bind. In the first case, where it is opposite, it cannot bind. The reason being, essentially here the positioning of this R in the presence of leucine, what essentially it helps is binding of arginine going and sitting to the sulfate group of the naphthalene, some of the naphthalene derivatives. So you can look at those druggable sites on the proteins which are specific, which evolved. You don't need to do it. The reason is, most of the times in our studies what we do is, I do a mutation, then it binds stronger, then it binds weaker. But the reality is, I take a protein in a in vitro tube and I show this residue is regulating the binding, weaker, stronger. But the thing is, how do you translate this in a human system? Since you published a paper, our protein is not going to change the amino acid inside, right? So that is why it is important to look essentially what changes are present, or you need to do at the gene level itself so that they can help. This is one of the example yesterday. I think Professor Chari has told how the cat forms with one single mutant and stuff. Okay, so now let's complicate the life. So until now we have only proteins. Let's go into protein surfaces. So that means we have more complexes. So what are the attributes or attacks of a given protein complex? Okay, so it is more difficult than the proteins, of course. Since the protein PPIs, protein-protein interactions are protein complexes, you can have hetero or homo-oligomeric, which I will show the examples. Then you have obligate, non-obligate, transient, permanent, domain-domain, domain peptide. You have so many of those. Okay, they can form in very different, and a protein complex can form between a structured protein and a disordered protein or intrinsically disordered proteins. So there is a hypothesis in the protein world that these nascent polypeptide chains are the intrinsically disordered polypeptide chains. Okay, they can work as a protein hub since they don't have intrinsically a structure. So I can go with anyone, with any partner. So I can get a little shape or structure as per my partner. So they can work as a hub protein, hubs, or so on and so forth. Okay, so this is what I am talking. We have domain-domain in the protein interactions. You have complementary surfaces. You can have domain peptide. You can have peptide-peptide, and so on. What do you look at when you have these protein-protein interfaces or protein-protein interactions? You need to see the size and the number of residues. When I am talking about the number of residues, I am talking about how many number of interactions engaged at the interface. I am not really worried about the size. Okay, so then what is the composition and the complementarity? So whether we have hydrophobic interactions, whether we have hydrogen bonds. Since if you see any of these cancers or any of these transcription factor complexes and everything, like when you are trying to interrupt the protein-protein complexes with small molecule inhibitors, right? Now we have so many libraries where you can scan these protein-protein complexes. Okay, these are some things which you will see, what type of pockets, what type of binding forces, or what are the secondary structure. If see the way you disrupt a coil-coil domain is far different that of you disrupt a beta-beta strand, or you have a simple interaction between tertiary structures, right? So the secondary structure beta-beta, anti-beta parallel, or a coil-coil is much tougher to disrupt rather than a simple 2 or 3 hydrogen bonds which are complementing each other. So that is why it is essential for us to understand these interactions. Now you can have the inhibitors, okay, you can have synthetic molecules, you can now have peptidomics, now peptides, natural compounds, you can have any of those. Okay, so this is where the ligand screening happens when it go to complexes, and of course you have to see whatever the ligand you are looking at, it is reaching into the cell where exactly this protein-protein complex forms. If not, in the real world, only in an in vitro tube, you can show with NMR or binding with a X-ray or something, but essentially it won't reach to the place where it is. Okay, now how do you target these interactions? As I told you, you can have a small molecule, you can use it. You can have other proteins or a peptide based inhibitors. So there are very classical examples. To be frank, rather than we tell scientists discovered this, I can tell you actually nature devised majority of this. The best example of hijacking a proteins to proteins is viruses. You take the corona or you take any of those, even the chemokines which I am showing, very cleverly the viruses make something called chemokine binding proteins. So instead of this chemokine go and bind to G protein coupled receptor, that protein hijacks. Then once it hijacks, you forget about any leukocyte recruitment in the body and the immune invasion takes place. So we can get lot of information. Essentially, the viruses can teach a lot for us how to target the systems. Okay, this is how you screen the drugs under a protein-protein interface. So you have a protein-protein interface. Of course, this is where actually all the things comes, and this is where no single technique can give you the full information. Okay, this is where essentially you need your cryo-EM, you need your X-ray crystallography, since you have macromolecular complexes. Of course, Mahavir will explain in the next how it is useful to have a combination of 2 or 3 different structural techniques in order to get a structural information of all these complex molecules. Okay, so you generate, then you do the high throughput screening. As I told you, can do the fluorescence based screening, then you do validation assays, microscale thermophoresis, ITC, SPR and everything. Then the hits and everything, you can use NMR. NMR is something which you can use in the identification, then you can use in the structural process and also structural optimization hits, what we discussed as SAR process, structure activity relationship. That is why people want NMR, since you can use it at multiple steps wherever you want. It comes as like okay, I am there, I can help you to a certain extent in this step also. Okay, so this is one good experiment. What people do using NMR is cross saturation experiment. Okay, cross saturation NMR is nothing but your STD NMR, but the only difference is here you don't have a small molecule ligand, right? So what here is, you are trying to identify the interface between 2 proteins. Okay, you have one big protein, one small protein. How do I identify what is the interface between these 2 proteins? So NMR can nicely identify this. So what you do in this, here you have one protein, small protein, protein 1, protein 2. Now what you do here is, the larger protein you don't do anything. Okay, you essentially keep the larger protein without labeling or something. So now you want to see on the smaller protein side what interaction. So what you do is, you do a 15N labeling. So you can measure the HSQC of this one, right? Then if you see this one, I wrote it as C2H. C2H, that means essentially we are doing a deuterium labeling for this protein. There is a reason to do a deuterium labeling. So what you do is, essentially now you put the selectively label the protons. Again, generally you do in the 1.5 to 3 methyl range. So you selectively excite these protons, and now you transfer these thing cross saturate to the ligand. Then what happens is, essentially the most worst properties for a vector to have is your CH vectors, aliphatic CH vectors and everything. So in order to remove those relaxation properties and everything, what essentially you do is, you put a deuterium, then the whole relaxation properties will change. Then this will be transferred and it will come at the interface, and what essentially you see is, whichever is interacting due to the differential phenomenon, you will see a attenuation of the intensities. Okay, so this is what is shown. You have a complex protein 1, protein 2. So now here there is nothing labeled, unlabeled, and this is deuterium labeled and N15 labeled. So you get a HSQC of this protein. Okay, so this is one of the example, the binding of insulin to its receptor. Okay, insulin receptor is very big and you have an insulin small molecule. So what you can do is, it's not essential that always you have to get a 15N. Okay, you can look at the, as I told you, chemical shift doesn't mean that always everyone will give you a nice spoon into your mouth with a 15N and proton, right? Chemical sheets of large molecules are far, far different. Sometimes you don't see, suppose if you take a protein of 70 kilodalton, 80 kilodalton, okay, you have 700 amino acids. Essentially, you will see 300 or 400 NHPs. The reason may be, even though you may have 700, but the overlaps. So in that cases, what people do is, essentially, they will do something called LBIAC label. They label the gamma 1, gamma 2 of valine, delta 1, delta 2 of leucine, delta 1 of isoleucine, and the C beta of alanine. So then they look only into those methyl signals. Whatever the perturbations you can see here, you can see there also. That is called methyl perturbations. So this is what in this example you are seeing, like, you know, this is a 13C ppm and a proton. You will see in the on and off resonance, what you will see is this intensity is going down. So specifically remaining all the amino acids which are not participating will be similar, but the ones which are participating will have the attenuation of the intensities. So in that way you will know what are the essentially the residues which are mediating these protein-protein interface interactions. Okay. So now if you go to this list, and one more thing, again these molecules cross saturation you will use when there is a weak binding and transient. You see, you always need this on and off, okay, then only things work. So there are several complexes which we have, people use it for these. Now I will show you one more thing in protein-protein complexes or protein-protein inhibitors. Now take a system, previously we have taken a protein complex with one ligand or a receptor and protein, and or a protein with binding sites, something, right? Now take it to a further. Instead of a monomeric protein, take a dimeric protein. Okay. So now what happens, what we want to do as a dimeric protein, I consider a homodimeric protein also as a protein-protein interaction. Since two monomeric units are coming here and forming a stable interaction between the two subunits, right? Now it can form homodimeric or proteins can form heterodimeric. Now upon addition of a ligand, you need a dimer or monomer. So if it is a ligand is breaking these two, dissociating, you will get two monomers, and it can associate into a dimer. The property of the ligand is defined by the quality of the protein. If I want to defunctionalize the molecule, suppose even in the homodimeric complexes, I will tell you, it is not needed, both the monomers are stable enough. The monomers can be partially folded, upon forming a dimer, they can get a state. Then the functional state essentially is a dimer. The moment I broke the dimer, this monomer is of no use. So then I have to dissociate. Now the second thing is, upon dimerization, the functional sites can be masked. So the monomer is essentially nothing. But the mask between monomers and dimers, this equilibria is something which regulates when to function, when not to function. What my work is, throw the full equilibrium to one side, either if I want a function or if I don't want a function. Okay. So you can look in those interactions when you have these multimeric proteins, dimeric, trimeric, tetrameric or something. What you can have is the protein ligands or the drugs binding at the interface. Essentially, when they are binding at interface and dissociating or associating the proteins, they are called direct ligands. Or the second thing is, they can bind at some of the other site and then also they can dissociate or associate. Then you call them as allosteric binders, right? So you can have allosteric binders, you can have direct binders, or you can have a binder in a dimeric molecule where it binds both the units and it doesn't affect the dimerization. So then you can call it as the dimerization and the binding are two independent events. We can have all these cases, and then once it is dissociating or once a ligand is associating the molecules, then again you have a beauty of NMR, like where you can tell when it is associating these molecules, what is it doing to the protein? Is it enhancing the stability? Is it changing the relaxation properties? Is it changing the dynamics? what essentially happens? All these properties, then you can use all the traditional structural and dynamic properties you can measure by using NMR spectroscopy. So this is one of the example which I already showed. These are the 3 chemokines, CXL 1, 2, 3. Essentially I will tell you these are the chemokines. Okay, when there is a injury or insult happens to the cell, these chemokines are produced, and now these chemokines will be decorated on the endothelial cell surface by using something called glycosaminoglycans, which is your heparin, chondroitin sulfate, any of those, okay, which are present on proteoglycans. Now they form a gradient here, and now on this below the endothelial cell surface you have the blood stream going, and what your blood contains? Essentially the leukocytes. Okay, now the leukocytes will see the migration of these. When they are seen, then when they see a gradient, then they know now we have to enter into the cell, there is some harm to the cell. So now the chemokine will be released by the glycosaminoglycans, bound to the G protein coupled receptors, then rest is all a story. G protein complexes activate and all the signaling events happen, then you have this one. So now these proteins, what they are good at is, they can be monomers, they can be dimers, or they can be higher order oligomers. So in some cases both monomers and dimers are functional, in some cases only monomers are functional, in some cases dimers are functional, and in few cases all the functionality is lost by forming a tetramers. Okay, so here we can show when you have multiple conformations. Now we are coming into the heterogeneity, right? If you have a dimer, what you can have is, you can have a full dimer or a monomer dimer equilibrating between 2 positions. When you have monomers and dimers under equilibrium, under slow exchange, then you can use the uniqueness of NMR to identify both monomer and dimer conformations at the same time. Okay, so you have monomers and dimers, you can see green and red, you can assign all of them, and now you have these monomers and dimers, now you add a ligand. Now the ligand is your heparin or glycosaminoglycan. So what I told, on the cell surface it has to put all the molecules together, right? So the GAG's essential role is put the oligomers at one place. More the oligomers, more the gradient and more the signal happens. So now you see all the monomers are vanished upon adding GAG, it forms dimers. Okay, so now these dimers, you can have the dimers, since you have all these 3 proteins, as I told you, evolved with similar conditions other than few minor amino acids, it can form heterodimers and homodimers. Now what GAGs does to heterodimers and homodimers, whether it only dimerizes homodimers or whether it does heterodimers or it is specific. So what you can see is, they do form, these chemokines do form heterodimers. You see now instead of 2 peaks of homodimer and monomer, you will see a 3rd peak of heterodimer also. And these GAGs essentially dimerize both homodimers and heterodimers. But all the GAGs cannot do it. You see, some GAGs will do it, some don't do it. The sulfation pattern is important. So there is rules. Like if you have couple of sulfation, if they are not in a proper position, they don't do it. The best example is the heparin, which is used for your anticoagulant, right? You have a pentasaccharide, this one with a proper functionality. If not, you don't get the function what you have. Those are called, we call it as small molecular heparins. Okay, enoxaparin, deltaparin, all which is given as an injection. Now, similarly, for all these molecules, the specificity is very important. Everything won't allow it to dimerize. Now, since it is dimerizing, you understand essentially it is stabilizing the dimer conformation. Now, can I get a dimer conformation? So, you can make these dimers are formed. So, you can make a dimer conformation. You just put a stable disulfide bond, and then by NMR what you can see is 2 sets of peaks now collapsing into 1 set of peak. So, that means you shifted all the population essentially into a dimer surface. Now, then you can measure the GAG interactions and you can see where the surface of these GAG interactions. You can map the surface, then you get pictures and you can get the FAT. So now you see, once you get a system ready, you can get all the information by using NMR. So now here you see, on these two helices, the gags are sitting on the top. Essentially what happens, these are the two helices. The moment these two helices moves like this monomer is apart, this monomer is apart. The moment I put a molecule on top of these helices, now these cannot go like this and it remains as a stable dimer. Okay. Now how do you make sure? Then you have to go back to your molecular biology experiment. You see, you know where your experiments are there, you know where your residues are there, you simply go back and mutate those residues which essentially show are participating in binding. You see, the moment you remove those 4 residues which are in binding, you see this, how nicely the peaks are shifting, and now you see no shift at all, it is dead. So it cannot change the equilibrium towards a dimer. Okay. What else it does? As I told you, NMR, you see, this is the helix. Now it quenches the dynamics of the helix. Since there is a motion, that motion is quenched, it changes. Now due to this, the gag is binding. You brilliantly see, like this one. In the case of dimer, if you see the hydrogen exchange, there is only a couple of residues which shows up. But the moment you add a glycosaminoglycan, all the residues comes up, are shown in a hydrogen exchange experiment. Hydrogen exchange means you put D2O instead of H2O. Whichever is a labile, they will disappear. Whichever is engaged in interactions, they will retain. So all the helix residues comes back only when you have your ligand present. That means ligand is essentially stabilizing your helix. Okay. And one more thing what you will see is, when you have these protein interfaces, complexes and everything, one other experiment which you like to do is your diffusion related experiments. So when you have monomers forming into oligomers, oligomers are going on. So essentially you do 2 or 3 simple experiments. One, you do a size exclusion chromatography, which is very common. Second, you will do a DLS experiment, dynamic light scattering, where you know, but you do a pulse field gradient PFG diffusion experiments. In the diffusion experiments also, you can calculate the diffusion coefficients and you can tell what is a dimer and what is a monomer. So you can find any oligomerization of these sort of things, you can in the protein-protein interactions or protein interfaces by using a diffusion constant. And this is the last example where you see you have monomers, dimers in a system, again dimer monomeric system. Now as I told you, I showed a ligand which shifts the equilibrium towards a dimer. Now this is a ligand where it binds away from the pocket but doesn't change the equilibrium. So that means if you have a monomer, one pocket, one ligand binds, you have a dimer, both pockets, both dimer binds, and it is away from the dimer interface. This is the dimer interface, it is away from the dimer interface, so it won't change. In such a case, what to expect? If you have such a case, what essentially you expect is the peaks, intensities don't change between monomer dimers since the equilibrium is not changing. Second thing is the affinity for a monomer and dimer almost remains same since the monomer is binding independently and dimer is having the same. It is not talking between the, that means the monomer dimer equilibrium is not coupled to the binding event. So that also we can check, we can see the binding which is different from that of the dimer interface, and now if you measure the binding events, then automatically you see all of them have same affinities. Okay. So this is where I will stop. So we have studied several of the examples under protein systems where dimers, monomers, how NMR used for small molecules, for large molecules, how you can identify multiple binding sites, so on, so forth. Okay. So some of these protein-protein interactions is there, we have compiled in this book. So I thank all the research group and Dinesh, where essentially we use lot of spectrometer time and all the funding agencies. With this, I thank you all.
Thank you, Krishna. So we are open for discussion. Questions?
Yeah, Ashutosh.
Just for that.
How do they study
see, if it is a very weak interaction, okay, so essentially in those millimolar range, anyway when you are doing titration, right, most of the time we use a millimolar protein. At least in the NMR case, we will saturate. But in the case of ITC or something, when we are doing this 50 micromolar protein or something, when it is in the millimolar, it becomes difficult, extremely difficult to saturate your binding interactions. Since in order to saturate, you have to go 10 times higher excess. But in the case of NMR, the advantage is even if it is 100 micromolar, okay, 200 micromolar. But these chemical shifts will come up since you may be using 0.3 millimolar sample, 0.4 millimolar sample. So these interactions is much easier to target, right? Since there may be very few interactions at the protein-protein interface. There may be couple of them, like strong interactions essentially have lot of, you see, the biggest target for those interactions is some of those transcription factors or some of those where they have these coil-coil sort of interactions, or secondary structures are involved in protein-protein complexes. There are two things. One is secondary structures which are involved. The second thing is tertiary, where it is like, you know, cup shaped, or there is a shape where shape complementarity is there. In the shape complementarity are those essentially which forms more of these interactions which are depending upon number of engaging atoms which are present in those surface, like number of hydrogen bonds or hydrophobic interactions. So this depending upon the, essentially you have to see which of the surface is having the these interactions and try to, actually we have these protein-protein ligand databases, okay. There are now for cancer and for several, there are so many databases. So now you screen those by giving some of the criteria like complementarity of one protein. Whichever you see, you can use one of the protein with more surface area or one of the protein where it can bind more of these molecules. You use it as a bite and you screen those molecules. And then you use that molecule which essentially now you put it. That is what your E2 complex, when you showed, I told you go to this protein-protein interaction database and you just check it. Yeah, if you have electrostatic, then only find some of these naphthalene derivatives or something where they have charged groups. So essentially, if you have when you have this interaction, on one side you have the lysine arginine and second side you have aspartic acid. Now you go to a naphthalene derivative or something where essentially there are sulfates or something and put it towards the one which has a positive surface. Then you can target.
Getting the signal of STD NMR, the reaction should be the first exchange after, and the other thing is that which was saturating the big protein should be greater than 10 kilodalton. If it is lower than 10 kilodalton, a smaller peptide in case of, then why we can't get the STD signal, or what happens there?
The transfer
all the gases gets saturated, and then we can don't get. But
see, you should have a enough size difference. You see, two things which we consider in the STD NMR is, one is the size difference, right? Even it is true for the cross saturation also when you have the two proteins. So what happens is, what here you are doing, you are having you are irradiating one and you are seeing the second one, right? Now if your irradiating molecule is so small, you are saturating everything.
Everything is getting saturated.
Yeah.
But in case of amyloid peptide, when it becomes fibril, then the size becomes small.
Yeah.
Then we can saturate the fibril and get the signal.
Then you see, so, yeah. Then again you see, if it has to work, it also depends upon the shapes also. See, when you take a protein, you consider it as a globular and sheet type, fibrils or something, right? So that also makes a difference, right? Essentially, where you are saturating. These are at the end, these are peptides, right? See, in the case of protein, what happens is you have continuous polypeptide chain, the transfer of signals.
Protein, yeah.
So here, what happens is these are intertwined by using the tertiary and quaternary interactions. So these some things you see on every, that is what, whether it is a cross saturation or STD, you have to work on a particular system. You have to optimize the ratio. You have to also optimize the irradiation on and off time, and also you have to make sure that which signals you are selectively irradiating that can transfer your signal.
Yeah.
Then only
we have
You
to
will
play
have-
on and off and see it is coming or not. Okay. And if in case that we are doing, we are previously doing no experiment. We have the first experiment in STD-NMR with a peptide and a ligand. The ligand can be also a small peptide or small molecule, anything. Then if we don't get any STD signal, then what we can...
you are expected to not to get, right? When you have both peptides or when you have peptide or small
Yeah,
molecule,
if it is-
essentially you...
Yeah,
If
small
you
peptide-
get, then it's a good thing. It's a wonder.
Yeah.
But essentially,
You
as I
don't
told you,
get
like
for
you won't.
peptide.
See, but these things, you see, when the other thing you see of, you see, when you are having the big protein and small molecule, the interface. But suppose if you have a small peptide, okay, 10 amino acid, and a small molecule,
Then
you can directly calculate the structure, right? Even you don't labeling, you don't need anything. Like you assign the amino acids, and you see even with a proton-proton toxi and proton-proton rosy or something, depending upon the size of your peptides, since nosy may flatten out. So you will directly get the interactions.
That is okay, but it is also 40 kilodalton, like 40 amino acid residues.
Okay, it's a little bigger protein. But the size is still, see NMR, always the NMR experiments, that is what I showed, like several set of NMR experiments. See,
Yeah.
every set of NMR experiments has its disadvantage. So you shouldn't work, you see, sometimes what people do is, people also work, you see, it's not like for every peptide nosy doesn't work, but rosy we do for the peptides, right? But now you are taking a 60 amino acid as a peptide. For 60 amino acids, I am a protein. So if you are out of the range, then the experiments will work. But now what you have is, you have like around 4 kilodalton or something, your peptide, and the small amounts. That is too narrow in range. And also with the peptides, the other problem which comes is, you don't have a full tertiary structures, right? Yeah.
That's the problem. Okay, thank you, sir.
Very nice talk, sir.
Thank you.
So my question is, as you said, ligand can be anything.
Yeah.
So can it be a nucleic acid?
Yeah, he will tell. Next talk is nucleic acid. So we go into protein nucleic acid for next 1 hour.
And can we also study nucleic acid and a small molecule interaction interface?
Yeah.
to this?
Yeah, that is one of the field. You see, when I focused on protein ligand, right? Now you take this protein, you can consider this as protein as biomolecule. Okay, then you can have carbohydrate. So whatever the heparin I showed, people have done this heparin as a carbohydrate, and then small molecule interactions, then RNA small molecule interactions, DNA small molecule interactions, then protein small molecule. You see, we have a matrix of 5 or 6 molecules this side, 5 or 6 molecules. Okay, any line is possible.
Thank you, sir.
SNPs in humans and mice, you showed one evolutionary tree also.
This one.
No, sir, the previous one.
Yeah.
Can you please explain?
Okay, so what here you are seeing is, so we have a protein family which are the growth chemokines. Okay, chemokines in humans are essentially like, we have around 50 chemokines. Okay, the highest number is seen in zebrafishes. Okay, there is a duplication of, there is a gene duplication happened, that is why. And what the hypothesis is, these chemokines are generally seen in the vertebrate systems, not below that. Okay, now in these also, it is expected that when the chromosomes are duplicated or the parts of chromosomes duplicated, they have evolved and they are duplicated, and then speciation. There are 2 events happening. First speciation can happen, then duplication can happen. Or duplication can happen and speciation can happen. So what we have looked into is, when we have the, see, essentially this is actually a backtracking for us. Okay, we have seen the surfaces are changing. Okay, and we clearly realize that these are the important surfaces where the GAGs has to bind, and they are changing. Now we went back, we did the sequence analysis, and we saw majority of the cases the sequence change when we have taken this all the 64 codons and we try to see manually what exactly why it is changing. When the K is going to E or K is going to N asparagine or whatever it is, right? So what we found is only one base which is going to change. Then we went back and looked at what essentially happened in the hierarchy of the whole species. Then we saw, like, they have duplicated, but there is a separate division between this side and the Laurasians, the primates and the rodents. But now, that is why if you see this diagram, essentially if you see, these are all CXL3, CXL2, CXL1. If you have 3 genes in the same species, you call them as paralogs.
Yeah.
Okay. If you have in different species, you have orthologs. So now, if you see in this one, this is Chinese hamster and everything, right? This is a rodent family.
Okay.
So in the rodent family, if you see, the paralogs are closer. For all the rodents, CXL3 will come one place, 2 will come one place, 1 will. And the same is true the primates also. You see gibbon, orangutan, human, you see all the 1s, all the 2s, all the 3s. Now, you go to the Laurasians, which is your cow, horse and all the animals. There, essentially the orthologs are much closer than the paralogs. So that means what we have defined is, that is why the mouse chemokines have closer properties to the human chemokines. The reason being, all these things are duplicated, then they have speciated, that's why they had not changed. But in the case of Laurasians, the speciation happened first, that is why.
First you look into the sequence SNPs, then after you done this NMR, right?
Yeah.
Okay,
let me tell you. Don't confuse. This, okay. We have no NMR.
Okay.
You completely do a evolutionary study, then you identify these surfaces, then on the basis of your hypothesis of these surfaces, then you know these are different surfaces, then as a protein chemist you know they should bind differently. Then you go back and clone these proteins and do all these NMR experiments and show those evolutionary whatever is there. So this is a work of like different pieces,
you
bring it together so that you get a logical conclusion.
So one more thing, in that tree you selected any outgroup for any particular outgroup constructed?
Whatever the sequences which are present in all these families, whatever the sequences we have incorporated. You see, the main problem is you have 3 paralogs, right? Essentially what is difficult is getting all the sequences of the paralog in a given species. We have done whatever the sequences is there, but someone, if they want to work more deep, they should go into genome and then they can extract the sequences. But see, essentially what it is as a structural biologist, I have a hypothesis this should be different. But at the same time, I have a curiosity how evolutionary it is done. That is why we stopped at that place. Then we moved ahead with all the NMR, heterodimers, homodimers and all the things. All these things of shape complementarity, that one amino acid changes. We got the clues from here. Then we never wanted to dig back this one. And then we moved how these changes, we used NMR and other things.
So I have one more doubt. If you are giving the protein sequence to some online portals like Swissport or something like that, we are getting a structure of protein which is similar to ours. So can we say what kind of structure, in what stage, like it's in the primary, tertiary or can we predict something like that?
Swissport,
yeah.
If you have a good homology, it will give a tertiary structure, right?
Yes, sir. So how can we confirm? It's not giving as such as ours, but it's showing somewhat similar to that.
Yeah, that is where you have to go back and see the protein families. You see, sometimes it is difficult, even in the yesterday's talk you saw, right? Still, that what Professor Chari has shown as I am Krishna, I will tell you. That is the work, one of the work done by my senior. That means in 2005, 6 or something. Still after 15 years, we don't know. You see, essentially why you will trust or I will trust. Archaea should have islets, but these are some proteins have more functionalities. Okay, it is forming a fold, but a protein fold can also have different functions. But unfortunately, we don't know the function of that one. Similarly, when you are getting a structure in a Swiss thing or something, essentially what you have to do is you got some structure. Okay, now you go back and look at to all the protein family, super families of this structure and see what the functions of these families.
Just 1 second to interrupt you, because what you're asking is actually the structure which you got.
Yeah.
means you can take it with a pinch of salt, okay, always. Okay. Closest being means it gives a fold only, never the structure.
Yes.
But the first thing is to understand the difference between structure and
Fold.
It gives only the fold. Okay. And fold also can be little, at best 0.5, 0.596, 60% accuracy. And unless you do experimental verification, it remains just a prediction. Alpha fold, as Charlie has said yesterday, is the closest thing which has come across recent years, last years, which is able to predict the structure to a high degree of accuracy. But still, there are outliers, and you have to experimentally verify and say, okay, this is structure. So prediction is a completely different thing.
Okay. Yeah. Thank you, sir. Thank you, sir.
Okay, so we thank Mr. Mohan for a nice explanation and given talk. And what we do, we go for a tea and snacks, and we meet again at 11:30. And that's a very interesting talk about nucleic acid with some of the really good work which Mahavir has done recently. Probably. So we'll listen to that. And after that, those who are interested can go their own samples. Somebody who has still their own samples for NMR and other things. I can help you. And then we'll give you the souvenirs, and then you are at your own. Right? After lunch, maybe you can have a lunch and then. So we meet again sharply at 11:30 so that we don't delay the proceedings.
after the tea and the final lecture, final oral presentation, and the interesting one which many people have more money is hardly been covered, the protein nucleic acid part. And it's a pleasure to welcome Mahavir Singh. He's a Delhi guy actually, and now he is in a much greener and cooler area, Bangalore, with that Ministry of Sciences. So from Delhi, he had a kind of in space zero quantum transition to Germany in the space, Martinstein, where he worked at with Haddock in Max Planck, and after that he moved to UCLA with Julie Figel, the famous AD who worked extensively with the protein nucleic acid structures and nucleic acid structures also. So he worked there and published some of the very great work there. And after that he returned back to molecular biophysics unit, which is one of the great places with synonymous with the structural biology in India. So and he's working there for the last few years. Now he's currently an associate professor there. So now I invite Mahavir to give this final lecture.
Okay, thanks Neil for this kind introduction and the invitation to be here in this and give this talk in this workshop. I titled actually my talk, it's Probing Protein and Protein Nucleic Interactions using NMR and their perturbation by NMR spectroscopy. Before the first talk, Krishna gave such a wonderful overview of different NMR method that one can use to study protein interactions and their perturbations by the inhibitors or the molecules, small molecules. So that was great because I will just it's sort of an extension of that talk where I will show you a few more examples of which directly emanate from my work, past work and the current work. And a couple of times Neil and Krishna mentioned that for nucleic acid can be also ligand for the drugs, right? But unfortunately I don't have any example of that where nucleic acid is targeted. Okay, I mean we study protein nucleic acid interactions and perturb those interactions, but nucleic acid themselves can be targeted. Okay, can be targeted in a different biological context. Okay, so when Neil asked me to come for this workshop, I told him that we are not directly working now on finding doing the protein drug interactions or finding a molecule to perturb a biological interaction. But then he said, yeah, just talk about whatever you have been doing in the past or here in Bangalore. So I thought, okay, I'll time travel and I realized that during my PhD I was actually involved in projects where we were actually doing protein small molecule interactions and trying to perturb very important interaction in the cell, which is between p53 and its antagonist MDM2. So I time traveled back and I have some slides. The first part of my talk will be I'll show you example of using how using simple NMR experiment you can study important biological interactions and their perturbations. Okay, so for the part one, I'll show you some examples from that work which I did, which I was involved. I mean, I was involved directly or indirectly at that time in Martinsried in Germany in Professor Tad Holak's group. So in part one, I am going to show you some examples from there, and in the second part, I'll show you some of our recent result on a project that we have in the lab, which is on bacterial toxin antitoxin system. And many people here, many people now in this field believe that they can be targeted for developing potential antibacterial drug. Okay, especially in this era of multi-drug resistance that is emerging. So okay, so let's go ahead. So so I think several of us would know this is a typical cell cycle. This is controlled by these cyclin dependent kinases which depend on this protein called cyclin. They control the cell cycle by controlling other proteins, and their concentration goes up and down in the cyclic manner, right? They are driving the cell cycle. But in cancer, right? And I'm giving you very general idea, because since then I have moved on to do some different thing. In cancer cells, basically, the certain checks which are there in the cell cycle are lost. Okay, and there is a protein called P53, which is maybe many people consider the most important protein. It's a tumor suppressor protein, which is a transcription factor essentially. It has a DNA binding domain, and it drives the expression of certain proteins which will inhibit the cycling, and so it will not allow cell cycle to progress in case of, for example, cancer cell, right? So P53 is considered as guardian of genome. Okay, so it's a very important protein. Now, you can the importance of P53 can be underscored by the fact that in almost more than 50% of all the human cancer, P53 is mutated. Okay, so loss of P53 basically causes the cancer progression. That is directly linked, right? Now, at that time in the lab, we were working on P53 MDM2 interaction. I'll come to this. Also, my main project was on another protein called retinoblastic protein, but I will not talk about that today. So, in further simplistic term, and as I told you, the NMR method and so on, maybe after 5 days of the conference, you would know what all method or what all experiment in NMR you can do to monitor protein-protein interaction. You take my talk as an example how you can apply it on some system. So as I mentioned, in several of the cancers, P53 is mutated, or P53 is kept in an inactive form by its binding to its antagonist in the cell called MDM2. Okay, there is a protein called MDM2. P53 tetramerizes. It has a tetramerization domain, so it is existing as a tetramer. Okay, and MDM2 binds and keeps it in the inactive form. In this situation, whether when P53 is mutated or is kept in the inactive form, you will lead to uncontrolled cell growth, that is tumor growth, right? So what you would want is free P53 from this. Of course, mutation is a different thing. You can't easily cure that, but you can release the wild type P53 if you disrupt this interaction between MDM2 and P53. And this had been, I think, still maybe many pharma companies, big groups all around the world, they have been trying to understand and trying to disrupt this interaction so that you can release P53 in solution. So you want this scenario where you release P53. So, sorry, I can probably use this. I'm not used to this. So, okay. So P53 is about 400 amino acid long protein. As I mentioned, it has a tetramerization domain. It has a DNA binding domain, and it has a MDM2 binding site here. Okay, the transcription activation domain we also call it. Right now. MDM2 is 491 long protein. It has different domain. It binds to P53, keeps it in the inactive form. Also, it is E3 ubiquitin ligase. So it also targets P53 for degradation through proteasomal pathway. Now, it has at the N terminal of this protein is a P53 binding domain. Okay, so this N terminal part is the P53 binding domain. Now, at that time in the lab, several of us were studying this interaction. So, we designed. I mean, we and other people designed a peptide which will just have the MDM2 binding site, and because purifying full length P53 was challenging. Okay, especially because of its tetramerization and so on. So just to study this interaction, as often in NMR lab we will clone the binding sites, okay, the binding regions or the binding domains, and study the interaction there, and then extrapolate it to the protein function. Now, and similarly from MDM2, we just made only the P53 binding region from the N-terminal domain, right? Now, this domain of MDM, the p53 binding domain of MDM2 gives a very nice, HSQC spectra. Now you must have seen this, throughout the conference, this spectra. So this gives a very nice spectra. You can actually assign it, which we did, and this is the assignment. Now we know each peak's identity. So at that time, in TATS group, they were working on a group of these compound called chalcone. Okay, they are diphenyl propane on derivative, and they found that they actually can bind to MDM2. They were working on several of these chalcones, right, and defining how they bind MDM2 and whether they can disrupt MDM2 p53 interaction. So Krishna beautifully explained the binding, the slow exchange, fast exchange, weak binding, strong binding in his talk. So it is then very easy for you to understand now here. So when we titrated now chalcone, one of the chalcone to M2 and monitor the binding, you can see here that some of the peak specifically perturb, right? So you have, for example, this glycine here is nicely going from here to here. Okay, this is an example of fast exchange, weak binding, right? You have a continuous movement of peak from the apo form to then the bound form. Okay. So from here, when you map it on the structure, the chalcone binding to MDM2, you can to the first approximation you can guess that this will be the binding site. Okay, this is a model, but these residues were maximally perturbed. Okay, when this particular chalcone was added to MDM2. Now we also understood how p53 peptide binds to MDM2, and if you see that the p53 peptide first through chemical perturbation and then by real structure binds like this, right? Now from here you can see that the binding sites are quite distinct. The chalcone is perturbing this region, whereas p53 is actually binding here, right? Now the main idea, so this is this is a chalcone, this is definitely a binder, but whether it will be able to dissociate this is questionable, right? So often this is what we face. We get a binder, but whether it is active or not, right? So either it should actually, in case of a protein with the activity, for example, enzyme, it should reduce the activity, or in case of protein-protein interaction, it should be able to dislodge the interaction. Then only it will be sort of do its job. Okay, so from here you can see that this chalcone is unlikely to disrupt p53 MDM2 complex. So this is what I just told you. Mainly, it's easy to identify a binder, but whether it will disrupt that protein-protein interaction is the question, right? So we then work further, and we devised a this is simple NMR that we were doing, and see whether we can have an assay where we can see the dissociation of bound p53 from MDM2. So for this, what we did is we instead of just working on the MDM2 binding peptide, we now have a p53 construct where we have a longer p53 construct, right? So about 300 amino acid, about 35 kilodalton construct, where we deleted tetramerization domain because it used to complicate our purification and so on. So we can make this 292 residue construct of p53 which will have MDM2 binding site, right? So now instead of a peptide we have a bigger protein, right? Now now if you titrate now MDM2 the p53 binding domain of MDM2 with this larger p53 domain, this is what we observe, right? This is now because again this you have covered, this now became a large complex, right? This is now about 45 kilodalton complex. It tumbles now slowly, right? And because of the relaxation, you lose the most of the resonances, right? Because it became a larger complex. Now if you throw now a chalcone and you want to see whether the MDM2 release or not, this is what we did next. And when you add chalcone, actually it does not disrupt the the complex, right, as expected, as we were guessing. So the chalcones are not able to restore the spectra of MDM2 here. Now at that time, and the other way around, if you have a MDM2 bound to chalcone, and if you throw P53 to it, actually P53 still binds. So maybe it is dislodging it or binding simultaneously, right? But our idea is reverse, right? We want to have our molecule dislodge this complex, not the P53 dislodge this molecule. So at that time, as I mentioned, this is a very important interaction in the cell, and several big pharma company and big groups were working on defining molecules which can disrupt this interaction. This is from one company, one pharma company. A paper came in Science, around that time, where they discovered a group of compound, and this was the best one, called nutlin-3. This is the full form. Basically, they have imidazole, piperagine, and phenyl, this complex structure. So they define nutlin-3, which can actually bind to MDM2 and activate P53 by releasing P53, right? So we decided to test this compound nutlin-3 in our assay. So again, this is free MDM2. When it binds to P53, you lose most of the resonances because now the size is increased. Now if you add nutlin, you actually restore the spectra. Of course, nutlin binds to MDM2. There are shifts compared to the free MDM2 here. But you can see that MDM2 is free, and it must have freed P53, bound P53. So nutlin was definitely a great molecule, which could disrupt P53 MDM2 interaction. Okay. So here, no, that's what I'm saying. This is the free form, and now this is nutlin must be binding. So these are bound to nutlin. So there will be shifts. But the idea is that you recovered the MDM2 spectra. This will be bound to nutlin now. So this is free, and this is bound to nutlin, and P53 must be free, right? So of course we were doing mainly NMR and this biochemistry. We were not really looking at the cell that time. Okay, in my time. Since then, they have done several other things. So at that time they just called this assay antagonist induced dissociation assay, or IDA. Okay. Now this is as Krishna was saying, it's not that for every interaction this kind of clean result you are going to get, right? And they were very lucky that for MDM2 P53 interaction this was working, right? So you could add a molecule and see whether it can restore the spectra of MDM2 and test the efficacy of that molecule. Now again another high profile paper came at that time in Nature Medicine, where they found another molecule which was foran ring based molecule. They call it Rita, okay, which they said that it binds to P53 and thereby blocks this P53 MDM2 interaction. This is H sometime for human they write H MD2, M for N, and this for human. Although they are I think identical in terms of amino acid. So they said that small molecule Rita would bind to P53 and block P53 MDM2 interaction. Again, so basically this is the scenario. P53, the Rita should bind to P53 and release both P53 and MDM2. So in our assay again we did the same thing. We added small molecule Rita, and we see that it actually does not dissociate the complex. Okay. Now that became at that time Tad wrote to the journal, say that in our assays it does not dissociate it. So we should probably, because taking these molecules people do so many other experiments, right? So we ended up actually putting our result in Nature Medicine. They accepted our our results, and we wrote that the NMR indicates that small molecule Rita does not dissociate P53 MDM2 interaction, right? And of course they wrote a rebuttal and so on. So that's a story. So then of course these were the known compound. In Tad's group they wanted to also develop their own compound, and so this is from my colleague at that time. So they screened a chemical compound library, and they found this isoquinone based compound as a binder for MDM2, and this particular compound for NX6 showed actually interesting result. So again they titrated MDM2 with this compound, they saw that it binds to the p53 binding site on MDM2, and in their dissociation assay, actually to large extent it can dissociate bound p53 from MDM2. So this was also just like nutlin. This was a compound which was also able to rescue bound p53 from MDM2. Okay. So at that time, as again I'm quoting Krishna again and again, as Krishna said, that one thing about NMR, however fast you are, but you still take you need more material, more sample of protein. Also, longer time. NMR is expensive and so on. So at that time, we just thought that whether by just recording 1D proton spectra, can we actually follow this interaction? Okay, because it will you will use less sample for 1D spectra compared to 2D spectra. You don't have to label your protein, and the sensitivity is also higher for just proton spectra. So again, you need a good probe. So what you are focusing here is the side chain indole proton of tryptophan. Okay, because if you follow other proton, there are n number of other protons, so they all come together. It is difficult to follow whether it is NH proton or the upfield other proton like the H alpha and from the side chain. So the indole proton, because it's around 10 ppm, it gives you its resonance. You can follow it. Now at that time, in the construct of p53 that we were using, there were 2 tryptophan. And in MDM2 there were no tryptophan, the construct that we were using, right? So this is now p53. This is when it binds to MDM2, one of the proton disappears, okay, which is around the binding pocket. When you add nutlin, okay, you get back the spectra of p53, okay. And same when you add NXN6, you get mostly the spectra of p53 back. Now if you can also integrate the peak and see what percentage of signal recovered. Here almost 100% of p53 was we calculated that is released back. Here about 70% of p were released. Okay, so nutlin was better, we concluded, and of course I am not showing you other data where we were also doing side by side like ITC and other measurement. So nutlin was a better binder and was able to dissociate this interaction better than NXN6. So we also wanted to monitor, of course, MDM2 because these compounds actually bind to MDM2, not p53. But in MDM2, as I said, there were no tryptophan. So first thing they did is they selected some of the residues which were on the surface, not leaving the binding site of p53, converted them into tryptophan. Okay, they connected tryptophan mutant of these. Now, so now you have MDM2 which has a tryptophan. Okay, and of course we check that the folding of this tryptophan mutant is correct. Their stability is comparable to wild type. We don't see the effect because of the mutation. Okay, so we checked all that. And we found that some of them actually are as good as wild type, and they can be used now for in our 1D version of these dissociation assays. So now this is MDM2 spectra, free MDM2 spectra. Now this at 101, I think, yeah, this was a threonine changed to tryptophan, right? Now. You can see if you now add nutlin to it, okay, nutlin, you get into, this is again a case of slow exchange, which again Krishna showed very beautifully in his slides. You can see the 2 peak for corresponding to maybe the free and the bound form, and when you add more of nutlin, you shift it completely to the bound form. Whereas in case of NXN6, the peak continuously moves to the bound form, right? So again, this spectra beautifully demonstrated that nutlin is a better binder than NSN6. Now, of course, this is again binding of MDM2 to nutlin, or nutlin binding to MDM2. We wanted to see whether through 1D we can also monitor dissociation of p53 and MDM2. So Here is an example through 1D also we could monitor it. So this is now p53 spectra, the 2 tryptophan of p53, right? This is the 101 W of MDM2, the free form. When you mix, these form complex. Okay, this peak actually disappears, and this peak is also broadened. So you see sort of this is this kind of spectra where it is the bound form of p53 and MDM2. And when you add the the nutlin 3, okay, these both the proteins are now free, right? So this this and this actually we concluded that they come at this position and give this this is the compound, of course, will bind to MDM2. So but it is released from p53, and this peak they come now at the same resonance, doubling the peak. Okay, so the intensity of this peak is higher because now it corresponds to both p53 W23 and MDM2 101. Okay. Again, we concluded that through 1D we can follow the dissociation of p53 MDM2 complex by nutlin 3. Okay, this is again similar example using another tryptophan in the which was engineered in MDM2. Okay, so at that time because this interaction, as I said, was so important, and several companies had their molecule to test. Tad actually went ahead and he opened a company just for studying this, a private company, and I think it was to some extent was successful. So they, people will send compound, and we test whether they can actually dissociate the p53 MDM2 interaction, or they are just the binder of either MDM2 or p53. So this is what we did, and I already told we could directly monitor the influence of ligand on protein-protein interaction on this very important p53 MDM2 interaction. They call it IDA for antagonist induced dissociation assay, and this could be followed by simple 2D HQC and 1D spectra. And up to where I was, there were several papers published. Since then, they modified and they went on doing many other things, peptide inhibitor and so on. Okay, for this interaction. Okay, so this was because I thought it would be relevant for this workshop, I digged and I thought I would present this part. Okay. So for this part, I must thank also it was wonderful to think back my time about 15 years maybe back when I was in Tad's group in Germany, and these were my colleague there who were working with me, and this was me working in that lab long time back. Okay. Then, as Neil said, I moved to do my postdoc at UCLA with Professor Julie Fegan. There we were actually studying telomerase enzyme, which is again a very hot enzyme for drug discovery. Okay, but I'm not going to show that work here. Again, in Julie's lab, there was no focus on actually developing drug. We were understanding the basic properties of the enzyme, its structure, and so on. Okay, and so then I joined MBU department at IISc, and there we are working on protein nucleic acid interactions, protein-protein interactions, and working on a couple of projects, a few projects. In one project, we are understanding the DNA binding properties of this array domain, which is relatively less characterized DNA binding domain. And in another protein project, we are studying how intrinsically disordered RGG region is able to bind G quadruplexes specifically. But I could not relate any of these projects to find a motivation for disrupting this interaction for the purpose of like discovering a drug and or inhibitor. But we have a project on something bacterial toxin antitoxin system, which many people in the field think that can have the potential to be developed as a means to inhibit bacterial growth and develop a antibacterial agent. So I am going to talk about this work now, and would ask you whether we can disrupt these complexes so that we can develop a molecule which will act as an antibacterial agent. So yeah, basically TA system is one of the mechanisms through which this TA, meaning toxin antitoxin system, is one of the mechanism by which bacteria defend itself from phage attack. Okay, bacteria are actually under constant threat of attack from the virus called bacteriophages, and we all know several defense mechanisms that bacteria has against invading viruses, including restriction modification system, CRISPR-Cas system. Okay, there are some other signaling processes and so on. But there is also one called abortive infection, okay, where the idea is that when there is a viral infection, before virus can replicate itself and make many hundreds and thousands of copies, bacteria will commit suicide, altruistic suicide. We call it abortive infection, so that it is the virus propagation to other bacteria is aborted. So TA system actually work here in the abortive, they are abortive infection system. So what happens? So in TA system there are two components. One is toxin, okay, and one is antitoxin. Toxin, and they are under same promoter. So this is the operonic system, right? So you have a antitoxin protein. It can be RNA, I will show you, and a toxin which is always a protein. Okay. Now this toxin is toxic for bacteria. If the bacteria expresses this in a free form, it will be toxic for bacteria. Okay. Why it is toxic? Because it's usually an enzyme, usually it is an RNase, okay, and in the free form it is going to cleave cellular RNA or bacterial RNA, okay, like your ribosomal RNA, mRNAs, tRNA, and thereby inhibit the cellular processes, vital cellular processes. So bacterial growth, either it will be arrested or bacteria will die. Okay, there will be a bacterial cell death. Okay, now so obviously bacteria cannot have free toxin. So how it sustains itself under this is that it makes a toxin, antitoxin, it antidote for this toxin. So you have an antitoxin which binds tightly to the toxin, forming this non-toxic TA complex, toxin antitoxin complex. Okay, and bacteria under regular homeostatic conditions will keep TA complex and live happily. There are biological functions assigned to TA system. One is that it helps in maintenance of plasmid. Okay, so many genes are actually kept on plasmid in bacteria, and the TA system is kept on those plasmids, so that you end up with a bacterial population which will have that plasmid. Otherwise, if you don't have that, the cell, after cell division, which does not have that plasmid, will also propagate. Eventually it will dilute, you will have both the population of bacteria. But here it will make sure that if you don't inherit that plasmid, this bacteria will die, and you will all the bacteria with the plasmid. Their primary role is actually against phage defense. Okay, so as I mentioned, when there is a phage attack, you have the TA system in bacteria. Several factors still sort of unknown, will be released from the phage, including of course its DNA, which will disrupt the TA complex. Okay, and in the free form, the toxin will be free, and it will kill the bacteria. So the phage will not be able to propagate to itself to the other bacteria in the colony. Okay, and the other role that is sort of assigned for the TA system is their role in the drug tolerance. Okay, antibiotic drug tolerance. Again, the same thing here. Under antibiotic stress, certain factors are released, which will which will inhibit the TA complex. Okay, but what happens? It does not kill bacteria, but it suppresses growth enough that they will go in a dormant state until antibiotic is there. And when antibiotic concentration is down, okay, conditions become favorable. Again, it will revert back and propagate. Okay, so we call this as a persistent cell formation. Okay, under a stress such as antibiotic stress. TA systems are classified into 8 types, from type 1 to type 2. I'll just give you some example of our work on type 2 and type 3 TA system. Okay, type 2 where both toxin and antitoxin are protein, and type 3 actually the antitoxin is RNA. Okay. So what as I said, in the free form, toxin inhibits bacterial cell growth, or that will put it in the dormant state, or cause the cell death. So and in normal bacteria, normal growing bacteria, it's kept as a TA complex, correct? So the idea is that if you can disrupt this complex, if you understand these complexes, and if you can disrupt this complex, this TA complex, toxin antitoxin complex, by a small molecule, by a peptide inhibitor, or a nucleic acid based aptamer, you can at will, in pathogenic bacteria, you can release toxin, okay, which will have a growth inhibitory effect in the bacteria. Okay. The other thing is, TA systems are completely bacterial stuff. Okay, they are not in eukaryotes. Okay, so if a drug is developed, I mean, it will have effect, sort of, right? So so people are thinking constantly about disrupt these interfaces so that you can activate toxin. Okay, especially that we have multi-drug resistance for most of the antibiotic now. We want to find other ways to kill bacteria. Okay, so I'll skip many of these slides for detail, okay, because we just come to the point. But for type 2 system, we have been working on a system which is called ICGEB-DBT. So type 2, as I mentioned, both toxin and antitoxin are protein. Okay, first you make the mRNA, you make both the protein, and the protein, it remains in a TA complex, which is non-toxic. Okay, and they are kept under same promoter. And most of the time, antitoxin is kept before toxin, because these toxins are actually very toxic for bacteria. So any small ratio towards toxin, and it will have a growth inhibitory role. So antitoxin is before usually in the operon, so that cell makes sure that antitoxin is before toxin. So when by the time toxin is made, you have antitoxin to neutralize it. So this is the system we were studying. It's called ICGEB-DBT. There is a toxin called ICGEB-B and antitoxin ICGEB-A. Here actually it is reversed. Somehow we can only speculate, but the antitoxin here is first, then the toxin. Okay. Now, yeah. So most of the operon, if you see, the antitoxin is first in the operon, then the toxin. So when you make it, of course, you are going to translate the antitoxin first. No, so it is going to make it, and first this will be made, then it will be made. I mean, there will be always the ratio, slide ratio towards antitoxin, because it is first in the operon, right? In case of type 3, I will show you, there is also, just to make it better, there is an anti-terminator in the middle. Okay, but here usually they are continuous. Okay, and to make further it is foolproof, because even in our experience, when we try to make only toxin, for example, it's very difficult, because these bacteria will not grow. Okay, when you are expressing only toxin. So what also happens is that the antitoxin has a DNA binding domain, and it binds to its own promoter operator sequences here, and auto regulates its expression. So basically it acts as a repression. Okay, so it will when you have enough of TA system, you don't need more, you shut it off. Also the operon its expression. So antitoxin has a DNA binding domain which will bind to the promoter operator region here in a specific manner, and thereby shutting off its own expression. Okay, so there are multiple ways through which the cells make sure that it doesn't kill itself. So basically we can't make toxin alone. We have to put them together as a TA complex in same vector, and we can then purify it. Okay, we purify it, the TA complex, and we get crystal. We solve the structure through. I don't know what it is now moving. Okay. So we solve the structure of this ICGEB-DBT complex through X-ray crystallography. This was too large for NMR. Actually, the NMR spectra there is no spectra here for this complex. It was totally broadened, right? So we saw this through X-ray methods. You can see this in the cyan color is the antitoxin, and this is the toxin. Okay, this is how it binds. It's a heterotetramer where two toxin are bound to two antitoxin, and antitoxin is dimerizing through this dimerization helix at the end terminus. Okay, and this region here is the DNA binding domain. It is a helix turn helix type of motif in antitoxin. This is how free antitoxin looks like. This is how free toxin looks like. It's a classical, microbial RNase T type fold. Okay. Again, I think I'll not spend much time here, but we saw that there is a lot of movement in antitoxin here. Okay, which we could also see through the movie, but maybe it's not playing. But doesn't matter for this talk. It doesn't matter. So as I mentioned. So, as an antibacterial strategy, what all can you do? You have the TA complex, okay? You can disrupt it by a small molecule. That is one thing. Several people are reporting there are problem associated with everything, but in principle you can disrupt the complex. There is also, as I said, the antitoxin has a DNA binding domain which binds to its operator and blocks its expression further, right? So you can also understand the how antitoxin binds to its promoter DNA, and then maybe manipulate that interaction so that the autoregulation process you can disrupt. Maybe it will also have inhibitory effect. Now, so we started doing this in this promoter region of ICGEB-DBT. There were these palindromic sequences identified. Okay. So, we know that for the highest specificity, typically DNA binding proteins will bind to palindrome in a dimeric fashion, right? So, for example, many restriction nucleases, many DNA binding factors and so on, domains and so on. So we did that. We took different palindrome. I am showing just one example here called PAL1, and we saw that the HIG A alone or HIG BA as a complex nicely binds to the palindrome sequences, okay, through this gel shift assay or through ITC experiment. Okay. Now we wanted to understand in detail how the DNA binding domain binds to the DNA. Yeah. Yes. 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 Yeah. So I mean, as for the thermodynamics, yeah, this is very interesting. So actually, you will this is a complex. So HIG BA complex also binds because DNA binding domain is free. Of course, to even when it binds to the toxin, this is there. So the TA complex also acts as a repressor. Alone, antitoxin also acts as a repressor. And yeah, these thermodynamic signatures are very distinct. So you would assume that this is mainly enthalpically driven. This is both entropy and enthalpy. You will essentially what we were asked this question when we submitted paper and so on. The only way you explain is that the number of water molecule released will be different in both complexes when it is binding as an antitoxin alone or when it is binding as a TA complex. Okay, so that will factor in the entropic contribution. So that's what I mean. Everything was speculative because DNA binding domain is free in crystal structure, and that is what is binding to DNA. DNA also is same. In this titration also DNA is identical. Okay, so the structural change that is happening must be releasing those water molecules. Yeah. Yeah. It remains in our whatever we have. The TA complex itself can bind stably. It does not dissociate. Okay. For several other TA system also this has been known. Okay. In some cases people have shown that it binds better as a free antitoxin. It binds better as a complex. All those but it's different for different TA system. Okay. But this observation, I mean, this is of course multiple times we repeated it and we consistently get this. We explained it through likely. Yes. All those. No. We ended up doing all control. Okay. We ended up doing all the controls. Yeah. Huh? Correct. So that was our aim. Unfortunately, we do get crystals, but they diffract very poorly. And maybe next time I'll show you. We have now some sort of idea from cryo-EM. But before that, because we also do NMR, we thought, okay, what can we understand through NMR? For that, we will have to simplify our system. This, this is the protein, our HIG-A, which is dimerized, which is a dimer. It gives a pathetic spectrum because, as I said, these two regions also move with respect to one another. Okay, so this actually gives a bad spectra. So we ended up, as we would do, we ended up doing just the DNA binding domain. So we isolated just the DNA binding domain, okay, which is a helix loop helix sort of motif found in other TA system, other protein, and we thought, okay, let us understand through NMR. So again, it now gives a beautiful HSQC, and we assign it. You guys all should be expert on this now, and we titrate it with the DNA, and we see nice chemical shift perturbations now, right? And if we map those perturbations, they fall on this, alpha helix, this loop, and this alpha helix, okay, this region, right? So based on that, we generated a, put everything that we often, many of us will do, we put it in ad hoc and generate a model, and this fits like this, okay. This mostly explains it, these palindromic sequences are here where, DNA binding domain will bind, okay, and also, the stoichiometry matches with our experiments such as SECMOLs and ITC and so on. Okay, so we have this model which we published. Right now we are also working on through cryo-EM to visualize this structure with the full protein. Right, this is a structure which is assembled, but based on the chemical shift perturbation data here. Okay, so this is where we are, and of course you take it the way you want. I mean we see that the largest perturbations are on the residue which are there in the market, but we use those perturbations to drive the model. So you take it the way you want to take. Yeah. So we did several HADAC. We keep DNA just free. We don't apply anything. We just say where the program docks it. In some cases, I don't know which model we are presenting here, but basically it will occupy the major group, right, where the DNA binding domains are majorly binding, and the other, sometimes we also say that this is the palindrome region, so this must be the binding site. So like that we also direct. Correct? Yeah. Okay, so part of this work we published I think now maybe 2 years back, in 2021 year, and some of these this is still not published. Okay, so as I said, why we want to understand this THC, the auto-regulation process is that these interfaces can be also perturbed, and we see that if you remove this auto-regulation, what happens? Okay, will it have a inhibitory function? Okay, so now I will quickly go to another type of TA system and see that that is interesting. Here, this is type 3 TA system where the antitoxin remains as RNA. Okay, so you have here. This is the operon, this is promoter, this is +1 transcription site. Then you have this sequence which is non-coding RNA. It will be made into RNA, but it will not be translated. Okay, this is code for antitoxin RNA. You have a row-dependent transcription terminated sequence here in between. Okay, and then you have a toxin gene. Okay, now the RNA is made, you get protein made. And this protein is an RNase itself. As I said, most of the toxins are RNases. So it cleaves. It will be, this RNA will be made like full length RNA. Let's call it. And it has repeats. So in this case, I am showing 1, 2, 3, 4, 4.5 repeat, which are here, right? So and this protein will cleave this RNA into individual repeat, and then assemble with one of the repeat to form this closed cyclic complexes. Okay, so we were working on this, and again, so we had a project, COE project, where we proposed to do this, and in E. coli, because at that time none of the type 3 system are identified in E. coli. Now last year, while we were writing our paper, this paper came from a lab in US, where they characterized first E. coli system in E. coli, did their functional assays. Okay. So we identified several actually in E. coli and so on, and we just through bioinformatics analysis we can group them into different clusters, they basically they have more identity for the protein sequences, also for the the RNA repeat and number of repeat, the length of the repeat and number of repeat are also unique for each cluster. Something like that is what we did through bioinformatic analysis with the with late Professor Srinivasan group in our department. So typically, when you want to now experimentally work on this system, what do? You go to the genome and you see all the signatures. So you have the promoter sequence, you have antitoxin repeat, you have this terminator sequence, which nicely folds into this hairpin loop terminator sequence, and you have the protein coding region. Okay, so then you clone it. In this case, we could not express this together. In this case, we had to modify our strategy where we had to put toxide RNA into separate plasmid. So you express both the plasmid and you see bacterial growth. Again, we are asked whether this is actually a TA system because we were just the predictive sequences. And of course, through growth assay, you can clearly see that when you express toxin, the growth is suppressed. Okay, and when you co-express it with antitoxin, again, it is restored. And you can also do through this colony. You can count the colony and conclude from there. What was interesting is that when we purify it, we actually get all the three. So you have TA complex, protein RNA complex, you have free toxin protein, and you have antitoxin. So in our ion exchange step, okay, we get three peaks: one for the free protein, one for the free RNA, one for the protein RNA complex from E. coli. Okay. Now this is if you want to do biochemistry, biophysics on the system, this is wonderful because you get all three species. Okay. The middle one we take, we purify it further, we see that everything is fine, we crystallize it, and also you can see here that you have the protein made and you have the RNA. Okay, it's a clear RNA. Okay, this is stain for RNA, this is stain for protein. And as I mentioned, from this strategy, we ended up getting all three of them, right, in single prep. Of course, protein is active as we have an artificial substrate where we see that it nicely cuts the RNA. Then we crystallize it, we solve the structure, and the structure looks like this. So you have three protein, okay, three toxin bound to one antitoxin repeat, forming this closed, heterohexameric assembly. Okay, this trimeric, a triangle-looking, heterohexameric assembly. And this is how the free toxin protein is, and this is how free antitoxin RNA is. Okay, so the RNA forms a pseudoknot structure in the middle, and you have the 5' and 3' end which interact with the RNA, in head to tail manner, doing this. So and here, when we see the RNA, this is a beautiful structure where you have several of these, three of these base triplets here. So this is actually a nice UU base pair. Okay, in the UU base pair, and this is a G coming to the major group site and further stabilizing this. So this is at the in the middle of this pseudoknot. Okay. Now of course we do through simple again NMR. We see that if you remove this interaction, we see disappearance of certain key base pairs. So why we want to understand the pseudoknot? I mean, what is the role of pseudoknot, right? That puzzles us. Why can't protein just bind to simply to the single strand part? Why you need this pseudoknot, right? Here I am showing you the 5' and the 3' end binding, right? So we think that for forming this closed complex, the pseudoknot is essential. So one way to understand, when we want to find a molecule to disrupt this, one way could be to destabilize these pseudoknot structures. If you find a molecule and find a base to destabilize pseudoknot, maybe this will disrupt also the binding here. Okay, so we are excited. I mean, there are ideas around how to destabilize it, because pseudoknot is a nice structure. It's often pseudoknot is at the core of folded RNA structure in many riboswitches and so on. So we think that if we understand the pseudoknot folding, we can actually maybe find also some molecule which can destabilize this. Okay, through mutations and all that, for the biological assays, people are doing it, we are doing it, but also finding some molecule like a metabolite, just in case of riboswitches, taking idea from there, how you can further stabilize or destabilize RNA structures. This is how it binds. I mean, if you see, align all the sequences here, of the toxin, the absolutely conserved residues actually form the R at the surface where the RNA is binding. Now, I told you this is an RNA and it has processed its own RNA. So this RNA is, basically, I think this may be 5 prime, so this is 3 prime. Okay, they are close enough. So protein has bound it and cleaved it here and got stuck to its own RNA. So for this own RNA, it's a sort of a suicide inhibitor. For the cellular RNA, when protein is free, when it is cleaving, for example, mRNA, it's going to cleave and release those fragments. Okay, but in case of its own antitoxin, it cleaves, but it's not able to get rid of it, and that is how it becomes an inactive complex. And you can see it's a nice cavity here for binding to this single strand part. So we also this is in detail, but from this complex itself we could understand its catalytic mechanism, right? So you have here 2 prime 3 prime the cyclic intermediate at the 3 prime nucleotide, which is often which is a intermediate in when you cleave phosphodiester bond, right? So it is well stabilized by different residues. Yeah, in the crystal structure, in all the we have a good resolution structure, we can see it nicely forming. So that will, when the phosphodiester bond is broken, you get through this intermediate, and ultimately in even in regular textbook, if you see, ultimately the phosphate will remain either on the 2 prime or the 3 prime after cleavage, but you have that intermediate formed in when you cleave phosphodiester bond. That basically what we are saying is that it's not that it cleaved the RNA first and then assembled with it. Most likely it bound to the larger repeat, cleaved it there and get stuck there. Okay, because otherwise we would not see this intermediate if it would assemble with the free RNA. Okay, so and then through that Michael Lau paper from MIT mentioned, right? Through their transcriptomic sort of analysis, they had predicted a consensus binding and cleavage site. So they predict the cleavage site like GAAU, which cleaves here, and this beautifully explains it. Actually, this is GAU only, and the cleavage here is between 2A. Okay, so this our data fits completely, explains their binding. So then. We wanted to understand, remember we also get free protein and free RNA, we wanted to understand through ITC how it binds. And this goes into this complex, right? From the cell we purify it. When we mix these two, we get this biphagic curve consistently in ITC, and we fit it to 2 binding events. Okay, so there is one binding event here and the other binding event here, which we think is because of 2 independent sort of binding sites. So although the protein is binding here, the 5 prime binding interface and the 3 prime binding interface are quite distinct. Okay, so this curve explains that. Okay, so again I am stuck. Yeah, so just to resolve that, we delete one end at a time, and we see I'll show you. So you can see there, this is the binding group for the 5 prime end. This is for the 3 prime end. You can see that the number of hydrogen bond and the surface area involved is different, right, for the both the sides. So that actually if you analyze your ITC carefully, you can support it from the ITC. When you actually delete one binding site at a time, you can make it monophagic, right? So coming back on NMR, so when we can purify all 3 of them, and this is RNA, and we know that making labeled RNA is very expensive. In fact, back in UCLA, we were making labeled RNA continuously. But here, I have not dared to make it because most of the time our RNA degrades. And in one time, if I order labeled NTPs and make it, and my student come and show that it is all similar, it will give me heart attack because in 5 lakh rupees or 4 lakh or whatever, 4-5 lakh rupees will go in me. So I have not been able to make labeled RNA here. But now, at least for this system, if we can, from E. coli, if we can purify RNA, I can label it, right? I can label it with N15, C13, at least, uniformly labeled sample we can get for this RNA and try to understand it through NMR spectroscopy. We have not done sort of it, I don't have a result, but you can this RNA at least you can get in the labeled form from E. coli. And we definitely can get RNA. This is the RNA that we get. You can see the immuno spectra. We match the walk, right? The hydrogen bond in the base pair region. This is how the complex looks like. This is about 100 kilodalton complex. We don't see many of the peak, right? This is too large. And when the free protein we record HSQC, this looks like that. This is also again not super good, but this is how the in NMR it looks like. Huh? Yeah, the oligomer, somehow it's I mean definitely not giving good HSQC spectra, but so far we have concentrated on solving the structure of the free form, bound form through crystallography. That had worked. But yeah, these things needs to be optimized further if you want to understand the whole process through NMR. And as I mentioned, now if you disrupt this complex, if we want to find a molecule, these I correlate back to my PhD work. We can use this spectra to find. Yeah. Immunoprecipitation. I know why would we do that? No, so we can purify this. No, I mean we get 3 distinct peak. We'll get the labeled complex, labeled whatever is. I mean all the nitrogen carbon will be labeled. We can purify the complex. We can purify the toxin free and antitoxin free. Yeah. Yeah, so RNA so far, heteronuclear part we have not looked yet. We have tried a couple of times. So what happens, sometime we'll get the free RNA nicely, and sometime we will not get it. Okay. So so far in the labeled prep, my students are not able to get enough RNA. So some tweaking is happening where we either get it or not. But this is the RNA. This is from one of the unlabeled LB prep. We get the RNA and we can see the RNA. Okay, but for the labeled prep we are now looking into it so that we can do some NMR on it. Right? Yeah, at least we'll get labeled RNA made from cell, if not through in vitro transcription. Okay, so yeah. This work, at least the structure part and all the initial characterization we recently published. And as I said, the motivation here is to find a molecule, okay, which can be a small molecule peptide or nucleic acid aptamer to disrupt the TA complex. Why we want to understand the autoregulation process or autorepression process is because that can also be perturbed, to see whether it will have a growth inhibitory role. And in case of type 3 TA system, I think the pseudonaut, if we destabilize pseudonaut, it may also have a effect on releasing the bound toxin. Okay. So just not us but the other people in this area, of course, are constantly thinking how to activate TA system by a peptide or by a molecule. Just as an example, I just read these 2 papers from a group. Okay, where they identified some potential candidate peptide candidate to disrupt the one of the TA system in MTB, okay, which belongs to VAB-BC class. Okay, so here VAB-B is toxin and antitoxin, and VAB-C is toxin. Okay. So what they did here, again you have VAB-BC toxin and toxin complex. They look at the structure. They design a peptide which is basically this yellow helix. Okay, here from the toxin, which they think should compete and release the bind to the antitoxin and thereby release the toxin. Right. So they take a peptide. They check whether it actually is disrupting. In their assays, it actually showed effect. So they have a simple assay here. As I said, these toxins are RNases. So you have a kit here. This is also commercially available. You have a RNA here, which is labeled with the fluorophore and a quencher. Right. So in the normal form, the fluorescence is quenched. But if you cleave it, the fluorophore is away from the quencher, and you will see increase in fluorescence. So if you incubate this with the TA complex, okay, you see that there is no enhancement of fluorescence, right? But when you disrupt it with the peptide, you can see enhancement of the fluorescence. So this using this assay, they actually tested their peptide. Further, they made them more potent, sort of by making them stable peptides. So you have these olefin linked amino acid, and through which you staple them, which increases their helicity, their structure, and their also uptake in the cell and so on, stability. And they show that the stable peptides are more potent. Okay, and through NMR, coming back on NMR, they showed that these peptides actually can bind specifically to the antitoxin. So this peptide is able to bind to this red guy here in the just a isolated peptide form. According to them, this is not our data. This I'm taking from that paper as a proof of principle. They are saying that the mainly peptide is binding where it should bind. Okay, so the residue here are perturbed here. So yeah, in this project so far, we were just focused on doing the structure, but going forward, we are designing some of our own compound and our own peptide, and maybe who knows, maybe in next meeting I'll show you some result. Okay. So yeah, in our TA group, TA project, these students Pankaj and Vikrant postdoc worked on ICGEB-DBT system, and this is Manikandan and Kavishri, they are working on, and Harshita, a new student, working on type 3, the protein RNA complex TA system. And this is my friend and collaborator in Tromsø, Uli Rothweiler, and my other collaborator on the TA project, right? And these are our fundings. So yeah, I mean, I definitely think that TA systems has potential. So hopefully we will identify some of the inhibitors. So thank you. If you guys have any idea how to, design an inhibitor for TA system, you can talk to me.
Thank you, Mahavir. Thank you. It's a very interesting work, actually.
Thanks.
So, although we have interrupted you quite a lot in between, but nevertheless, if you have some questions, yeah.
It's good that my talk was at the end, because it was not about NMR method, but you can use NMR to study these.
Hello, sir.
Yeah.
Nice. So my question is, like these RNA molecules, they already have 13p NMR active UTM. So cannot we use that for some RNA studies? Like
I'm sure you can use it. Yes. Yeah. Definitely. 13p, 31p NMR, right? I mean, Krishna again. It's good that Krishna gave talk before me. I can refer back to his talk. Correct. That will be difficult. Yeah. So if you see 30 spectra, usually they are only average 1 or 2 peak, right? Because the dispersion on the phosphate is less. Yes, absolutely.
Okay. Yeah. That's why we have to label it also 15 and 13.
Then you can get residue level. Yes, if you do that. Yes.
Thanks.
Yeah. Yeah, I think the main problem is we have to, but first we need to work to get good spectra, In the free form itself, these proteins are not giving good spectra.
No, I'm not asking protein like in free form. In the complex form, even in the microcrystalline or crystalline
form. Okay, yeah. I'll have to learn something for that. I see. Solid state. Okay. Sure. Yeah. Yeah, we can. I see. Interesting. Yeah.
ISC has a solid state setup also, no?
Yeah, we have a solid state machine. Unfortunately, it is not working right now. It is one of them that is actually quenched, so we have to revive it for some time. Anyway. Yeah. No, it should be revived soon. Yeah.
Anything more? Have any questions? Yeah, there are lots of in between already.
Sure.
Yeah. So thanks a lot, Mahavir.
Thank you
for coming all the way. And
thank you. Thank you. Krishna, एक photo लो. This is.
Yeah. It might have to open it soon.
Thank you. Oh, you have a photograph. Okay, thank you guys.
Thanks, Mahavir. So now we finally come to the end of this workshop. Theoretical part, lectures, of course. So I think some of you had some sample or something which wanted to run oil or something. So you can still have, I am here and my colleagues are also here. You can get a sample. Okay, that's okay. Then it's fine. So, but if you want something to do, means you can still go and do something. If you want to run something or spectra or something, do other work. You are welcome to do. In the evening, that's what. And of course, tomorrow, Sunday, is completely free for you all. And it has been a wonderful stay, I hope, for you guys. We tried our best to do things and revive some of the things which were frozen for last 2.5 years. There may be shortcomings, some of the shortcomings, and I really apologize for the certificate issue. I'm sorry, we could not do the things. Because of so many things happening, and all the mechanism has to be really revived for the of course ICGB is quite professional in running the workshops, and we have been running till 2019, but for 2 years everything had come to a standstill, and so it took time, and probably we will learn from what mistakes had happened this time, and we'll do much better next time. And so I think that the next course of workshop which are coming up in the 11 more in this DBT sponsored workshop, those things will be improved in a sense. And if you have any comments, critique of how to improve things, feel free to mail us. Of course, we have given you the feedback form, so you can fill it. And if you have some specific, you can write to us directly. And because that way we evolve. I mean, nothing is, nobody reaches a perfection anytime. So it's always a way to evolve and improve. So that's why I see the things. And I hope the foods and other things would have been good, and guest house also, it has opened, and probably after 2.5 years you all were the first occupant of the guest house. The rooms were actually made open for you guys. And so I'd like to call the people who really worked, and most of you have met. Tenzin, please. So Tenzin was a real. Please come home. So she worked almost for everything, the manual and all the things which were going all around. She did more major of work. Anupam, there, he designed the cover page and logo, all of what you see. Dr. Nidhi Verma, he worked also behind the scene for many other things. Maybe stand up and show your face. Priya is there, so she PhD student. Sonam, and she is there. So they all are my colleagues, so we try to work together and then try to get the things moving now. And of course, the few people from administrations are not here, like Tina, behind the scenes, all the mails which were exchanged with you people, Tina and Sachin were doing. Sweta did the major work of getting the administrative thing done. And the person you see behind the glass, Mang and Ravinder, had an excellent, provided the IT support. So all the lectures were actually live fed on YouTube, and these lectures will remain on YouTube. So anytime if you want to go back and see those lectures, you can go back and see them. And if you have query, all the mail IDs are there. And if you search any of the speakers' name, you will find their mail IDs. And you can mail them if you have queries, or if you want to collaborate further with any of the speakers. Each one of them have different expertise, coming from some metabolomics, coming from some hardcore drug discovery, protein nucleic acid interaction, protein-protein interaction, solid state NMR, or dynamics like Thenthil. So all have different, so if you have some problem, specific problem, you can write to them, collaborate with them, and that's the way the science moves, with the collaboration of different issues. One cannot do all alone. So with that, we'd like to thank you all again, coming here, being courageous during this tiring times, testing times, and many of you who have come from abroad. I know how it means going to the embassies during this time, really it's a big challenge actually. It means getting the visa, flight booking when the flights are costly, quite costly, not only because of the pandemic but also the war, which has made things quite costly. So if you have come, and it's a great thing, and I hope these spirits will remain high in coming days, and we'll move forward for excellence in science, excellence in working together, and for betterment of our everybody, the humanity as in the large. So thank you all, thank you, and after lunch, you can go to lunch, and that's all. So, and then you are free to roam around. And if you are moving around a Delhi, always carry a bottle of water, because it's 45-degree outside and practically no humidity, so you will not feel that you are getting dehydrated. So you will not feel. So carry enough electrolyte, because we don't want you to really get fainted on the street or something. But that's it. It's a global warming or whatever you call it. It's there. So thank you.